Okay, well, the title of our talk is there. So the origin curve, 1 becomes 2, 2 becomes 1. So the floor is yours.
Oh, thank you. Thank you, first of all, for the invitation and for all of you for coming to yet another Zoom talk. That would have not been my preferred format. I wish I was there in person. And I've also never been to India, and it's been my wish for years. But I've recently had a small baby, and I think she would not have taken it well if I remove her source of food and take it to another part of the world. So apologies for being in this 2D, but I hope we can still have some sort of a discussion. And if I usually tend to present my research kind of as a question and things we get to solve using or think we get to solve using our models. But I tend to leave a lot of details out. So if I leave anything out, please either ask or drop me an email later on if you want to know something. Okay, so I'll tell you a bit about our recent stream of research on shaping of cell membranes. We put in this biblical context, but it's really about discussing how you divide membranes, how you merge membranes. And I'll start with our, I guess, general motivation, which is cell division. Of course, it's the foundation of life. And we all come actually from one cell that divided a long time ago and kept on dividing. So in that sense, we're all one colony of brothers and sisters. But to me, being a soft matter physicist, it's really a physical process of how you take a soft droplet and make two. And you do so by producing mechanical work from within. And you might imagine there's like a million different ways to do so, but actually, not really. Across whole evolution, it's been done by protein filaments that assemble and at the same time disassemble, which is equally important. And they're coupled to some sort of deformable compartment. And of course, they need to consume energy to have this assembly, disassembly process. And really, how you do that, how you divide cells across all tree of life, there's really three main ways, and they're compartmentalized according to the branches of the tree of life. So this is the tree of life. It has two prokaryotic branches. So those are organisms that are single cells and don't have internal structures. One branch is called bacteria. You know about them, they make us sick. And the way they divide is they build a ring in the middle made of filaments called FTZ filaments. They're short and stiff. I'll play it again. And they treadmill, so they kind of run around the belly of the cell. And because bacteria is pressurized and quite stiff, they probably don't produce much mechanical force to squish it. But we know they guide the synthesis of a wall in the middle of the cell. So really the way bacteria divides is by building a partition in the middle of the cell. Then there's another prokaryotic branch of the tree of life called archaea. And they've been recognized as a separate branch of the tree of life only kind of in the late 70s. And the way they divide, at least those kind of evolutionarily younger archaea, so they're quite soft. They're the closest to like a vesicle that we have. And they have, again, a ring, protein filament in the middle called the escort filament. And it's long and flexible as opposed to short and stiff in bacteria. And it actually squishes the cell into it, produces mechanical force through some active geometry change that's driven by energy consumption. So those are these two prokaryotic branches of the tree of life. And then, for a long time, we thought eukaryotes, which is us. So you ping me again if it happens. I don't know. I have not seen anything on my side. So I'm not sure what's the last bit you've heard, but I was trying to say that for a long period of time, we thought eukaryotes were a special branch of the tree of life. But now we know that actually there's archaea that has proteins that were until recently considered to be eukaryotic specific. And through kind of a lot of work in the evolutionary biology in the recent years, I think now it's kind of accepted that eukaryotes are kind of just a twig of archaea. But they've been formed through symbiosis of archaea and bacteria. where bacteria went to become our mitochondria, and archaea gave us our cell body, our nucleus, and our outer membrane. And because eukaryotes are much bigger, so we still use these escort filaments that archaea use when we are squished to about 1 micron, which is the size of archaea. But because we are much bigger, we had to invent an additional mechanism on top of it, which is an actomyosin. You might have heard about it. And it squishes the cell. It's again filaments, this time driven by motors, which is an eukaryotic addition. It still squishes the cell to about 1 micron, after which the escort filaments that we got from archaea kick in. This is kind of the overview of cell division machinery across the tree of life. Today, I'll speak about archaea and bacteria. We started a bit recently working on actomyosin, but I will not talk about it today. And I'll just briefly comment on my favorite recent subject, which is the symbiosis between archaea and bacteria, which gave rise to eukaryotes. So we know that by the type of machinery we have in our nucleus and outer membrane, that archaea gave us our membrane reshaping machinery. And it's believed that this merging of archaea and bacteria occurred such that archaea kind of grew tentacles around bacteria and other archaea. So this is this blue thing growing tentacles around symbionts. And then you see, if you now merge these outer pieces, what you get is something that looks like a eukaryotic cell. And that's another line of research we've been recently playing with, kind of how can you get such a structure through a symbiosis of a soft cell with other cells. So this is kind of how people think it occurred, the big growing tentacles that then merged to give rise to eukaryotes. And it used to be kind of a theory, but in very recent years, this field of evolutionary cell biology experienced a boom, where two groups, a Japanese group and a Viennese group using a sample from Slovenia, managed to grow a symbiosis of these archaea that have eukaryotic specific proteins, so this kind of evolutionary younger archaea, and bacteria. And indeed, the way this symbiosis looks like is it has these long tentacles that are believed to be a parent of our internal membrane structures. So it's, as I showed you just earlier, these tentacles are then meant to become our ER, for instance. So now this is an extremely active field of research. And the question we also ask is, why, to make a symbiosis, would you want to make tentacles? So this is basically what I'll briefly speak about today. I'll speak first about the division of archaeal cells by protein filaments that squish the cell in two. Then I'll speak a bit about the formation of the bacterial division ring. Again, that ring does not squish the cell, but it guides the machinery of the cell wall that's being built. And finally, I'll speak a bit about these tentacles and the morphology of symbiosis. I'll first thank everybody who did the work. In our group, we do only computational experiments, basically, so theory and computation. This has been done on archaeal side by Anna, Lena, Sharon, Billy, and Tina. Chris has done all the bacterial work, and Maitane all the symbiosis work. And all the biology comes from Buzzbaum, Aurelia and Rue in Geneva, Sonia, Albert on membranes of archaea, and Seamus and Martin on bacteria. So I'll also mention them as I go. So let me first speak about archaeal cell division by this escort filament. So the name is a bit funny, escort, but for our purpose is a protein filament. So what Buzz managed to do in his lab in Cambridge is he managed to grow the closest archaeal relative to our cells that can be cultured in the lab. It's called Sulfolobus, and it comes from Yellowstone National Park. So it lives at like pH 2, temperature of 80 Celsius. So it's an extremophile. It took them, a couple of years to build a microscope that can live image these cells. And this is what he got. So this is the cell. It has divided its DNA, which is in blue, somehow, we don't know how. And then the way the cell divides is it builds a protein filament in the middle, which is in pink, which then templates another protein filament, another escort filament, which is in green. And together they keep put until energy is consumed, which is used to chew up this bottom filament, this pink, and then the green one somehow squishes the cell. So us being kind of polymer physicists of metaphysics, the way we thought about this is, you have one filament, one polymer, which probably matches the cell curvature, is kind of stiff and it stays there. And that filament templates another filament, which is now green. But that green filament really wants to have a smaller radius of curvature. So when it grows on top of this template, it's going to be stretched, kind of like a loaded spring. And then the purple one holds it in place until energy is consumed to remove the bottom one. And really in this kind of non-equilibrium self-assembly proteins in the cell, usually energy is consumed actually not for assembly, but for disassembly. So here energy is consumed for disassembly of the purple one. And then this green one, that wants to have a smaller radius, is released and will squish the cell. That's how we imagined it. So kind of to test whether this is possible in a physical system, we build the cell, basically a vesicle. And that vesicle is made of 1 bead thick membrane model. And then we bind a filament on the inside. Again, we don't have structures of these filaments, at least not the archaea, but for us, it's just a polymer. It's just made out of beads and springs. And these blue beads of our filament attached to the membrane via non-specific and a Lennard-Jones type of interactions. And then initially this filament matches the cell curvature to model this initial filament. And then to model this idea, when ATP is consumed, that the bottom filament is gone and the top one wants to go to smaller radius of curvature, we suddenly change the radius of curvature of this filament from a large to a small value, which is what we do by adjusting the bonds between these beads.
Can you hear me?
Now I hear you back.
Okay, perfect. Go on. Sorry.
I don't know what's it. Sorry, guys. And can I just make sure? So I have until 12:40, right? Is that okay?
Yeah, it's okay. Everything is okay. Just can you launch?
No, I'll say. I wanted to say I have time until 12. I have time until 40. I just want to make sure that I'm within time.
Yeah, don't worry. All the stops. Don't worry.
So I'll adjust the session that you get. You're not here forever. Anyway, let me show you what happens. Warn me when it happens again, the stopping of the sound. So what we do here is we take the cell and the filament on the side, we suddenly change the curvature, and it really does squish the cell. But of course, the filament being there, it still glues the two cells together. And if you go back to the experiment, you do see that the protein content in the cytoplasm goes up when the cell constricts, meaning that the filament disassembles. And when we stop disassembly, when Buzz stops disassembly in the cell, you see that the cell doesn't divide because the filaments stay there, glue the two cells together, and you very nicely see the two filaments separating by the curvature. So the blue one is constricting in the middle, and the one that has larger radius curvature is on the outside. So it sort of supports the hypothesis of two filaments with different curvatures. Okay, so we realized we need to start disassembling. But if you disassemble too quickly, it's really as if nothing happened. And in these non-equilibrium businesses, you need to do things with the right rate, with the right protocol. So we realized that if we disassemble slower, when the filament has had time to squish the cell, then you can get a cell nicely to divide. And here we didn't do anything to cut the neck. It's really when the filament is gone, the neck is very curved and the fluctuations happen. So we were happy about that, but what I thought was really valuable in this interaction with experiments is that now we can start directly comparing to experimental data. So we got a bunch of cells that are dividing. We measured their neck as they were dividing at time. And we quickly learned that cells are living organisms, they have a bit different sizes, they have different time to divide, but if you scale their neck with the initial cell size and the time with the time it takes to divide, all the experimental curves fall onto the same line. which is great. You know, as physicists, we love that. There's something inherent controlling the physics of the system. And in our simulations, the same thing happens. Like all the curves of this neck, as it's dividing in time, follow the same curve. It's just not the same curve as the experimental curve. And you know, that's anyway a more interesting scenario when your simulations don't match experiment, then you start learning something. What would be fun if everything just matched from the beginning? So what you see in experiment is that the filament has started to contract, but the membrane has not reacted for some time. And then there's almost like a linear decrease followed by a tail off. And in simulations, because we change the bonds everywhere at the same time, we put a huge amount of force in our filament at the same time, there's a quick initial squish of the cell, following by plateau. That made us think that probably we should also change the rate by which we change the curvature of the filament. We should not curve it everywhere. And it anyways makes sense that in the biological system and our curving of the filament really models the disassembly of this template. It makes sense that in biology that doesn't happen instantaneously. And also, if you go back to actually the way our filament looks like, I told you we're going to change the shape from a ring into a tight helix. But what actually happens is a bunch of these inversions of chirality, and that's a buckling instability. And it's the same one as that happens when you coil your Christmas ribbon. And you know, if you do it quickly, the system will get trapped in some sort of metastable state where helices of different chiralities meet, and that's, you know, that's energetically unfavorable, but the system is trapped. But if you coil your system slower, your ribbon slower, the system will have time to relax and you'll get a nicer helix. And, you know, I had a lot of discussions with our biological colleagues. They told me, well, this might be the case at the macro scale, but no nanoscale system looks like super coiled helix. And, you know, that's not true. And even the escort filaments, not the archaeal ones, because we don't have structure, but eukaryotic escort filaments that people have made structures of and report to helices actually have a lot of these inversions of chirality. And we thought they might be functional to help shorten the filament and make it contract. And also we think that, you know, you can control the number of these inversions of chirality by the rate of shrinking, basically by the rate by which this template has been removed. We go back to simulations and we tried a lot of protocols, because now again, you can think you can coil with a certain rate from one end, from both ends at random spots. And we realized that the only way we can get experimental curve, and we do it without any fitting, is if the filament coils at random positions, which mimics the way these ATPase choose out the bottom template. And then if you do it at random positions, which distributes tension the most uniformly, you get exactly the same curve as experimental one. You also get the system to divide extremely reliably because the tension is uniformly distributed and symmetrically. And this also makes predictions, right? If you change the rate by which these ATPase use up the bottom filament, you should change the slope of this curve. You should be able to change the shape of the filament. So these are kind of predictions we can give. And we have some recent data of from super resolution microscopy. The membrane is in red and this constricting filament is in green. And you might see that there are some kind of bumps going out of plane, which could be these supercoils of the filament. But I do think this going back between experiment, and this is real live cell experiment and simulation, is a valuable way to kind of try to start thinking of how the machinery of life might be working. So I'll just wrap up. I've showed you here, you know, with some interruptions, that active changes in filament composition. So you have multiple filaments, you remove one, so you change the composition of your composite filament that changes the shape of the filament. And that change in shape is transferred to the membrane to produce force. And it is really a different type of force productions as opposed to actin and tubulin, which consume energy in the single monomer level. Here you have a whole nanoscale structure that reshapes to produce force. So it's a bit of a different way of force production. And I kind of like this angle about learning about physics of life by studying it across evolution, because as I tried to say just before I got cut off, our cells have the same machinery, our meaning eukaryotic cells, to also do division, to do also trafficking. Like for instance, HIV budding uses this same machinery, but it's much more complicated in our cells. It has many more filaments, many more components. So going back to its evolutionary origins, where it's supposedly simpler in composition, but the physics should still be the same, I think is a fruitful way forward. So now I meant to comment a bit on the membranes of archaea, but I think I'll skip that in the interest of time. And I'll go to the other branch of the tree of life, so which is bacteria, which divide in a different way. So they still have a filament in the middle. It's called FTZ. And as opposed to being long and flexible and having geometry changes, it's actually full of short and stiff filaments that treadmill. So they grow and shrink and kind of run around the belly, which is what this movie was meant to, this simulation was meant to show. And I'll talk a bit about that. You know, to physicists, it might look the same. It's still a ring in the middle of the cell, but actually the way it works probably is quite different. And as I said, it's made of these lots of small, short filaments, and they grow and shrink dynamically. They grow on one end, shrink on the other end, and that makes them look like they're moving, running around. Okay, so I'll show you a movie. Maybe you see it, maybe not. So this is fluorescent. This is the cell. Fluorescently labeled is the protein, this FTZ protein that forms the ring in the middle. And it's initially fuzzy, and then it condenses into a ring and cell divides. It's kind of fuzzy, condenses, cell divides. Fuzzy, condenses, becomes bright, and then the cell divides. And so this condensation of proteins and filaments in a ring is a key step for the division, for the synthesis of the cell wall to happen. And we know that this, for this condensation, for the formation of the ring, the filaments need to treadmill. So they need to grow and shrink dynamically. If you put in a drug that stops treadmilling, the ring will never form and cell will never divide. So I'll speak a bit about this initial part. How does the ring form, this ring in the middle of the cell? Why do filaments need to constantly grow and shrink, right? They spend a lot of energy doing that, constantly growing and shrinking. And why do you need that to assemble a ring? We don't know how this ring then couples to the synthesis of the wall that divides the cell eventually. So I will not speak about the actual division part. That's still an active area of research, and we just got an 8 years long grant to study this. So hopefully in some number of years. But I'll speak about this initial part, the assembly of the protein filament ring and why the filaments need to treadmill. Okay, and bacteria is great because, you know, they make us sick, so they've been studied for so long. We know for this filament, we really know almost all the kinetic parameters. So we can build a simple Monte Carlo model for treadmilling of a single filament. So here, because bacteria is stiff, we studied it actually in 2D. We unroll the cell onto 2D. And then we can model the formation of this filament, the FTZ filament. Here I'm showing you just a single filament. So it nucleates. So we put a monomer, it nucleates, and then it grows on one end and shrinks on the other. So you add a monomer on one side, remove a monomer on the other side. To do that, I mean, the cell uses GTP. So what happens is the GTP gets hydrolyzed at the monomer-monomer interface anywhere in the filament. And when such an interface happens to come to the end of the filament, the monomer detaches because that interface is not as favorable, so monomer detaches. And again, we know all these rates, but basically the main and the only ingredients you need to get this process to work is you need to nucleate the filament, grow on one end and shrink on the other end with the same rate. And then it looks like the filament is propelling, like it's moving, but it's really not. And we know from both the in vivo and in vitro experiment that no monomer is moving. It's firmly bound to the surface by a very stiff actually linker, but it's adding and removing monomers, and it looks like it's moving. But it's very important for like collective phenomenon. to model it correctly. So not as a propulsion, it's something that moves, but as a growing region shrinkage. So I'll show you what happens when you now put many of them in the system. Filaments nucleate, grow on one end, shrink on the other end. And you grow, shrink, grow, shrink, grow, shrink. And after some time, you get them to align. And you measure the nematic order, the filaments align. So it's, again, seems like your favorite kind of active matter filaments pushing against each other. But again, nothing here is pushing, nothing here is moving. They're just growing and shrinking. But it kind of looks the same. It gives you the same type of an alignment. And that does, seem kind of relevant because we know that you need this growing and shrinking to get alignment and formation of the ring in vivo. So if you go now to in vitro data, and we here teamed up with Martin Luzek group here in ISTA that have high-speed AFM of this FTZ filament on supported lipid bilayers. And here they filaments nucleate on the membrane, they treadmill, they grow and shrink. And after some time, what you get is filaments start aligning. Very similarly to what we get in simulations. So now these are aligned filaments. You can actually compare them, and low densities and high density, they align. If you stop treadmilling by stopping disassembly in simulation and in experiment, you'll see the lack of alignment. So they cannot align if they don't grow and shrink, and they form these sort of defects, both in simulation and AFM. And actually, if you go to AFM and measure nematic order and compare to our simulations, so AFM is in these crosses, simulations are in colors, you get the data to fall into the same curve again without any fitting. So we really do get the same type of ordering in vitro experiment and in simulation, but we can tell why. And again, it's not because the filaments are pushing, but it's actually quite a simple mechanism. So what you see here, if a filament grows at an angle against its neighbor, which is here in purple. So what I was saying is that filament nucleates, and when it grows at an angle against the neighbor, it cannot grow, but it can still shrink. So what you get is that all the filaments that grow under angle, stochastically against another filament, cannot grow. So over time, you'll get that only those that grow parallel to each other will survive. So it's kind of the survival of the parallel. And by this kind of stochastic growing and shrinking, with time, you'll get that all the filaments align. So it's kind of a different form of active matter. It's not an active matter that propels, but an active matter that grows and shrinks. And because of that, you get an order. And then again, to compare to live cell data, we need to form a ring, and we need two more ingredients. So that ring needs to go perpendicularly to the cell axis. And for that, you need some curvature. And we know that these filaments, in particularly when they're bound to their membrane anchor, are quite curved. And because they're curved, they like to go along the curved direction of the cell as opposed to the non-curved one, which would be axial. So that's one ingredient you need, and this is what we add to our model. And then you need to somehow be able to form a ring in the middle of the cell. And we know that in the cell, there is a reaction diffusion going on underneath that prevents filaments to nucleate and grow on the cell poles, but effectively renders the nucleation and growth faster in the middle. These are the two ingredients we add. So basically, it's the curvature of the filament and the fact that the nucleation rate is faster and the growth rate is faster in the middle. So when we add that, so filaments are fuzzy, and then we kind of impose this chemical gradient. The filament, because it's constantly growing and shrinking, quickly reacts to this chemical gradient and forms the ring in the middle. And then that ring, I'll play it again, runs around the cell circumference. And we actually did this simulation at the exact density that has been measured in the cell, and it forms some sort of a patchy ring, just like it's been observed in super resolution data in experiment. But now we can go back to live cell data. Oops. And there it has been measured that the ring width is kind of, the ring nucleates very quickly. So its width decreases very quickly, sub-minute. But then the ring density grows very quickly, very slowly over tens of minutes. So it's kind of opposite to classical nucleation. It's fast nucleation, but slow growth. And this is exactly what we see in our simulation, and we could explain. So the ring nucleates very quickly because it reacts to this chemical gradient around it. And that's why this growing and shrinking is very useful, because it allows you to react to a gradient, to a chemical signal, right? To not grow where you shouldn't and grow where you should. But then the ring density goes up very slowly because you need to select stochastically against misaligned filaments. And that's a slow process that takes over 10 minutes. Okay, so I'll wrap up this bit too. I've hopefully shown you that this dynamic growth and shrinkage of filaments leads to collective alignment. And again, that's a very different type of active matter. I'd say it's an active, like turning over matter. So it consumes energy for shrinkage. So it grows and it consumes energy to shrink. And because of this process, it's able to align. And then to form a ring, you need the filament curvature and the underlying chemical pattern. So, people might ask, well, the ring might align just by diffusion. Actually, we know they don't diffuse. They're so firmly attached to the cell, but also the time scale would be much, much longer. You can also say that crosslinks between filaments might align them. We know in the cell there are crosslinks, but actually they just make the filament tighter. So again, that you will need diffusion for that process. And again, nothing diffuses here. So, and we know in the cell, the ring cannot form without treadmilling. So you really need this turnover to form alignment, self-organization. And then the question is whether such a ring can produce any even minimal force before the wall starts growing. And that's something we plan to explore in the future. And then maybe in the last 5 minutes, I'll just tell you a bit about this weird shape of cells in symbiosis. So I told you that eukaryotic R cells came to be through a symbiosis of archaea, these soft cells that somehow shoot out tubes and form symbiosis with bacteria, where bacteria went to become our mitochondria later on. So the question we ask is, and these are this symbiosis that has been altered of this Asgard, archaea, and other archaea bacteria, and they have these tentacles. And the question we ask, why do you need these tentacles for our symbiosis? Like, if I wanted to make a symbiosis with you, wouldn't I want to hug you completely? Why do I want to shoot out tentacles? And there's a bit of actin in some of these, but not fully filled. Also, they don't have motors. So we started to think, can you form tentacles simply by kind of different types of interactions between a symbiont and a host? And it really, what I thought was kind of a key change in thinking for us is to realize that these cells make a symbiosis to exchange metabolites, right? I give something to my symbiont and the symbiont gives something to me. But what I give to the symbiont is very different than what the symbiont gives to me. So this interaction is not reciprocal. Let me give you one example. We know that the symbiosis occurred roughly when the level of oxygen on Earth started increasing. And we know that this archaea is anaerobic, while bacteria is aerobic. So one hypothesis is that bacteria locally depletes oxygen because it's aerobic for archaea. So that would be the case where archaea wants to be close to bacteria because bacteria is depleting oxygen for it. So it wants to be in its environment. The bacteria might not care about archaea. It might not care that somebody is following it. Right. So this is a type of non-reciprocal interaction. I like somebody, but somebody might not like me back or might not care. Yes, I wanted to say that this is similar to what has been done to like colloids in chemical gradient, when one colloid releases chemicals that another colloid wants to follow, like in a diffusophoresis. But then another colloid might release different type of a gradient that the first colloid want to follow. But these, interactions don't need to be symmetric, right? So the way one colloid responds to chemical of the first one is different than the first one. The way the first one responds to the gradient of the second one, right? So that has been done in the colloidal context, both in experiment and, let's say, Raman Goldstein in simulations. And what you get is that one colloid follows another one, and the gradient of another one, you get something that looks like, predator-prey, so one chasing another one. So in our mind, we wanted to now put that a similar type of non-reciprocal interactions that mimic chemical exchange in membrane context. So just to remind you, if you have a particle, let's call it a symbiont, the blue particle, interacting with the cell in a equilibrium situation, what you'll get is, particle adheres a bit. If it interacts even stronger, it's going to get wrapped. If it interacts, much stronger, you'll get some sort of a budding, like a complete wrapping. So that's a completely A likes B, B likes A situation. But what if you break the symmetry now? So what if the symbiont wants to follow the gradient of the cell, but the cell does not care about the symbiont? So what we see in that case is you create kind of internal tube, like an invagination that's very dynamic. It kind of a bit grows and shrink. But, it's a case where because the symbiont wants to follow the cell, it creates kind of a point force, and the memory responds to point force by growing a tube. Now you can do it the other way around. The cell wants to follow the symbiont's gradient, in which case, if this gradient is weak, you'll deform the cell a bit. But if this gradient is stronger, you'll see that the cell wants to follow the symbiont, and it does so by growing a tube, right? And it's simply because, there's effectively, again, a point force happening here, and the membrane responds to a point force by growing a tube. Can you hear me? Or it has happened again?
We hear you perfectly. So.
Oh, perfect. It's just for me, it started being silent. Okay, and then these are really the last couple of slides. Now you can do many symbionts interacting chemically with the cell, and then in this case, you get many tubes that grow. You can also, depending on the number of symbionts, you can get all sorts of non-equilibrium shape of your cell, all sorts of like dancing dynamics. This is all unpublished preliminary data, but it shows, something that we are excited about at the moment, and that's interactions due to chemical exchanges between symbionts at the cell. And why would you form tentacles? What would they look like? And then hopefully comparing, which is something we're currently doing with BuzzBAM, to shapes of cells in symbiosis. So I think I'll wrap up here. Yes, the point is membranes, it's diffused a bit, and that the membranes get shaped in response to chemical gradients, which might be important in the context of symbiosis, maybe in the context of evolution, but we are taking evolution as an inspiration to try to study the physical mechanisms behind the chemically interacting cells. Okay, and I'll again thank my team. I mentioned them in the beginning, those who did the work, everything we know about archaea, evolution, and escort we learned from Buzz. Martin Seamus, in this case, taught us everything we know about bacteria and FTZ. I didn't have time to speak a bit about escort work we did early on and membrane work we did with Sonia. And I'll just say that we are advertising some postdoc positions in Vienna if you want to live like an empress with us, or that's how we imagine this. Please, or if you know somebody, please get in touch. And I'll take any questions if that's possible.
Great. So thanks a lot. Very, very refreshing talk. So any questions?
Hello. Hello, can you hear me?
Yes, hi.
Yeah, very interesting talk. I had a question, most of these processes are, my question is relating to the FTEZ ring. So maybe I've missed something there. You said, it's a thread milling process where, you keep adding these monomers at the front edge, and then, you delete at the other end, right? That's the kind of thread milling that you spoke about. But I mean, is there no role of active processes in these things, in these kind of phenomena? And how does the force get generated? Like, how do these cells, divide into two?
So in FTZ.
Yeah.
Yeah, so yes, exactly. So they treadmill, as you said, add a monomer and remove a monomer. But it's unclear, or there is no strong evidence that produces any force. But we do know that this, the cells, bacterial cells have cell wall, this kind of strong polymer shell. And we do know that cell wall synthesis occurs at this point. So basically, synthetic machinery of the cell wall attaches to this ring. And the way they divide is that somehow the synthesis of the cell wall occurs at this ring towards the, radially on the inside, and then you build a new material in the middle and you partition the cell into it. This is how they divide. How exactly that happens mechanistically, we don't know. If that was your question.
Yeah. Okay. So I got the answer to your to my question. So the second thing was so except in the last part where you're talking about this reaction colloidal particles. So in the first part also you did not mention the role of any of the active processes. So many of these things are biophysical phenomena where I would expect that activity would play a role.
Oh, for sure. No, for sure. It's an active process. So I said that it's two rings, right? And then they stay together unless you consume ATP. So there's an ATPase that comes and chews up this bottom template. So that's an energy-driven process. You need to have energy, right, to
how are these implemented in the simulations?
Exactly. So we just disassemble. So basically, this energy is used to disassemble the first filament. And to model this idea that the disassembly releases a second filament that has a smaller radius of curvature, we suddenly change the curvature of the filament. And that's an active step, right? So you can do it, which we have also done. You can put two filaments one to another, one that wants to have smaller curvature, and the first one, which is much stiffer, and it keeps it there. And then you just remove the first one, and the second one goes to its preferred radius of curvature. But just a removal of this first one, or effectively change in curvature of this second one, is mimicking an active step, like a chemical change.
Okay, fine. Yeah, thanks.
So in a sense, these are all chemically active systems.
Any other question? No? I have one. So it is known which is the mechanism for starting the process, which is the rings that activate the division. Is it a mechanical trigger or is it a chemical one?
What triggers the removal of this first one?
No, the question is at some point the division starts by the activation of those rings, and which is the mechanism that triggered this process? Is a mechanical one that the cell is too big, or is a chemical process that activate the beginning of the process?
Yeah, so that's not clear. So once the ring has been formed, the second one forms on top of it, and then the first one is gone, and the division happens. It's all kind of quick. But what tells the cell to form the first ring? It's unclear. So there is a nucleator, but then what tells the nucleator to go there? Right, we don't know. We now do know that it's coupled to the DNA division, because these cells somehow divide DNA, but they don't have spindle. So it's also unclear how DNA has been divided, and it's known that this is coupled. So when DNA has been partially divided, then this nucleator comes and tells them.
I was thinking it was just the cell has a really big size, and the mechanical signal that was triggering.
So you thought it's some sort of like a matching between the curvature of this filament and the cell size, basically. It could be. Okay. We've been talking about doing these experiments under different osmotic conditions so that you can change a bit the cell size. But yeah, we don't we don't have the data yet.
And exactly the same question for the end of the process. For instance, the end of the alignment. What triggers that is the end? Is any chemical signal, or is the physics of the alignment that say is done, we stop?
So what tells the ring to form there is really the chemical signal, which is this underlying chemical pattern, like in E. coli, it's called the MINS system. I mean, but there are in Bacillus it's like nucleoid occlusion. But basically, that's believed to be how the cell measures its size. There's an underlying reaction diffusion going on, and there is like a standing wave when the cell is of a certain length, and that's effectively what that prevents nucleation of the filaments and the poles and tells the ring to form in the middle. So it's a chemical signal, but then alignment happens through this treadmilling. So we know that if you do have this chemical signal, but prevent treadmilling through a drug, you do get filaments there, but they don't form a ring, they don't align.
Okay. Any other question? Well, thanks a lot. Donc, let's thank Andel again.
Thank you so much, and for your patience with.
Thank you.
Thank you so much, and many greetings from Vienna.